Good morning, everyone. My name is Sarah, and I am reporting on breaking news of a crime that occurred late last night in Ithaca, New York. The police were called at approximately 11:05 p.m. All they could hear was a young woman hyperventilating, saying that a man was dead in an alley. The police tried to get more information from her, but she had only discovered the body, not witnessed the crime. It took 11 minutes for the first police car to arrive, and when they got there, it appeared that the man was stabbed in the stomach multiple times, with the murder weapon nowhere to be seen. The forensic analysts, Erica and Amanda, arrived on the scene next and noted that there was a struggle between the victim and the killer, as blood was found under the victim's fingernails.
This blood came from the killer, who must have received a cut in the struggle. Leaving behind blood can be used in our investigation. Several suspects were identified from nearby bystanders.
It was time to get started. In order to solve this murder mystery, we must take advantage of DNA profiling, which is when you analyze DNA from samples of bodily tissues or fluids. No two people, except for identical twins, share the exact same DNA profile. To find the murderer, we will take the blood sample collected from the crime scene and compare it to the DNA profiles of each suspect.
Since we only have a small sample of blood, we will use the PCR technique to amplify the DNA with it.
Having the exact same DNA profile as another person is very rare. Because of this, scientists like Amanda and Erica can use techniques to separate DNA strands for comparison of crime scene and suspect DNA.
In the case of a crime scene like this, the first thing we do is find short tandem repeats in the killer's DNA from the crime scene. Short tandem repeats are non-coding DNA, meaning they do not encode protein sequences and are not phenotypically expressed. Microsatellites are named after their number of short tandem repeats.
So now we have our first poll question. What is the name of this microsatellite with repeat motif of GATA?
I know you're counting. So the question is, I will jump back to the picture so you can see it. What's your set? There are about 10 more seconds. Most students answered 7 points.
Yep, so the correct answer was 7.2 because there were 7 repeats of GATA, and at the end there was a GA sequence, which counts as 0.2 rather than 0.5, which can be a misconception. So it's important to keep in mind that each extra nucleotide after the full sequence is going to count as 0.1. So in this case, we have 2 extra, so 0.2. And so in conclusion, this microsatellite is named 7.2.
Once we have found our short tandem repeats within the DNA, a polymerase chain reaction is run to amplify these sections. We do this with the help of primers, which are fragments of either RNA or DNA that serve as a starting point for DNA synthesis. Next, the enzyme DNA polymerase targets specific primers which indicate STR loci. It attaches to them and amplifies the DNA from this point onward. The new strand detaches from the template and becomes its own new template for the next sequence of replication.
Now you may ask, why is it important to replicate DNA? It is important to increase the DNA of the target sequence to a working quantity. Basically, if we don't have enough strands of the target DNA, the results will not be adequate. Once the supply of DNA is large enough, gel electrophoresis is used to separate the DNA fragments by their size and charge.
The separation of DNA fragments in a gel electrophoresis is what will help us determine the similarities between the genetic information of our murderer and of our suspects. First, we make an agar gel for which the DNA will pass through. Next, a buffer solution is added to the gel.
Okay, so now we're going to have another poll question to lead into talking about gel electrophoresis. We noticed that in the lab last week, we poured a buffer solution over the gel in our gel electrophoresis machine, and we're curious to see what you guys think was the purpose for that. So a lot of you guys are right with a lot of your answers shown. The buffer solution provides ions for the conduction of electric current that allows the DNA to move through the gel electrophoresis, and it separates the strands based on size due to charge. The DNA is combined with dye and added to the slots in the gel on the anode side of the charge machine. Gel electrophoresis. DNA is placed on the anode side because DNA itself is negative and it repels from the anode and attracts toward the cathode. Over the course of an hour, the fragments of DNA will move across the gel, and the small fragments will travel the farthest. From there, we can visually determine which suspect has the most similar short tandem repeat sequence as the DNA found at the crime scene.
So this leads us into our next poll question. Who do you think committed the crime based on the results we got in our gel electrophoresis?
I will start the question and come back to it.
Okay, so the correct answer is suspect 2, because the pattern of the DNA strands on the gel are the same as the crime scene DNA. So as we've learned, each person has a unique short tandem repeat number, and so...
Just to show you, he doesn't answer correctly.
And so the shorter bands with the fewer short tandem repeats will travel the farthest, and so that leads to the differing patterns shown here. And suspect 2 matches the crime scene, so we can say that suspect 2 is likely to have been the murderer.
Since we now know who the murderer is, we will inform the police of our findings. Suspect number 2 will remain in custody, and our gel electrophoresis DNA data will be kept on file to be presented during his trial and sentencing.
Now that we have identified the guilty suspect, we will go into briefly discussing ethics related to what happens to DNA once it's collected. The question is, who should be in charge of storing DNA, not only from crimes, but also from people who immigrate to the United States? DNA is collected from immigrants for purposes of identifying family relations. The question is, which is our next poll question, should DNA be held by private firms or governments? So there really is no right or wrong answer to this without getting into politics, which we won't get into. So it's good to keep in mind that if the DNA is held by governments, these databases can be securely monitored and regulated. However, the authoritative power it gives governments can be concerning and give way to corruption. Alternatively, if the information were held publicly, privacy would be difficult to maintain, even if firms could create an open database for sharing data.
That's it. Thanks, guys.
Thank you.